free extra points for your grade. So go into modules, biology, modules. We're in week 6, February 8th through 12th. Today is the 9th. We're going to go into that bell ringer. Caleb, you need to do these with us. All right. When you get here, click on Submit Assignment. We're going to do this together. So if you have yesterday's notes, you can get the definition and look at it for some of the answers. So homeostasis. So those that were here, what can we put for homeostasis? Keeping what stable? Think about a fever. If we have a fever, our internal temperature is no longer stable. So homeostasis means keeping internal environment of an organism stable. So that could be pH, that could be temperature, keeping the internal environment of an organism stable. And the main organelle we talked about yesterday, we had a little reading about it. What was that organelle? The cell what?
Mitochondria. Wait.
Not yet. Starts with cell.
Diffusion.
Well, it's about diffusion. And diffusion is a process that it uses.
Cell membrane.
So that's the main one involved in homeostasis. And then an example of how our body maintains homeostasis based on what we talked about yesterday is the movement of materials into and out of the cell. You could put, that's the main thing we talked about yesterday. That's what we're going to look at, is getting rid of materials that we don't need to keep that, like waste materials and things like that. And then 2 main types of transport. We're going to look at one of them in more detail today. That was toward the end of the notes. One starts with a P and one starts with an A. We have passive transport we're going to look at today. And then tomorrow we're going to look at active transport. So by doing this is 4 extra points that will be submitted to your grade. So get that typed in.
Okay.
Then we're going to look at passive transport, and then I'm going to, those that are here, you're going to set up a little experiment, and I'm going to model it. So Amber, you'll just need to listen and write down any information that I give that you might need for it, and I'll explain when we get there. All right, so everybody hit submit.
Wait 1 second. 1, 2. Mhm.
Joshua. Yes, sir. Are you good? I can go out on it.
Yeah.
You good, Amber? I can submit.
Yeah, you can submit.
Okay. All right, then you're gonna go back to modules. Interesting, it was, I'm gonna submit it. All right, cell transport notes part 2. So that's what we're gonna do, for the first little bit. And then we're gonna begin this lab on osmosis, and so we'll go into here, we're gonna do something with it each day. And there are 2 videos, and then we've got a practice sheet, if we have time, that we'll work on. So let's go into the notes first. You got notes yesterday. If you were here, it's gonna be the back of the first page. If you, were not here and you're doing it online, they should pop up right under here. I've got to get down to where I'm at in here. So we're gonna start with passive transport for those notes. And then if you, weren't here yesterday and you're here today, there may be times you can back up to that first part. If you've got, what's going on? Your charger? Well, we're done with the computer right now, so let me message and see if anybody can send a charger down.
Sure.
Does anybody have, oh, you have a charger in these? Just set it over here, or if you can.
Got that big bubble cut on, bro.
Oh, that's gonna be hard. Just set it over here. and-
Talk to you. Oh.
I'm talking to both of y'all.
Yeah, she can, my camera's wide enough, she can.
She's a whole. That's why you sick.
I'm not even
Oh.
sick.
Okay, so,
For no reason.
I ain't want to go. I didn't feel like being late to school.
all right, diffusion, passive transport, passive. If you are passive, you are not doing or using energy. So if you are laying around at home watching TV, you're being kind of passive. So diffusion is one of these methods of passive transport. And so molecules, if you think about if somebody's sprays cologne or body spray, perfume at one end of the room, eventually if you're at the whole other end, you're going to smell it. That's diffusion. So these molecules are going to be all lumped at one end of the room, and they're going to be moving around and spreading out to where there's less of them, so that over time, you would have sort of an equal amount throughout. So what we call this is going down a concentration gradient. You're going from lots of it to a little of it. And these brackets that are square represent the word concentration. It's kind of a shorthand for concentration. If you think you can remember that, you can use it as a shorthand. Gases tend to do this, like oxygen and carbon dioxide are 2 examples. So this dashed line here represents the cell membrane, and these small molecules are able to diffuse through the membrane, through those phospholipids we talked about yesterday, and move from an area of high concentration to an area of lower concentration. And over time, you get it balanced out where you have equal number on both sides. So that is diffusion. Josh, were you?
Yeah, I got
You're
it.
good? Okay. Facilitated diffusion is the same thing, and if you read the article yesterday, it mentioned this. This is sort of a helping diffusion. You've got to have a helper. So this is a transport protein that is a channel. It has an opening through it, and it allows the diffusion of larger molecules or molecules that can't go through the phospholipid because they are hydrophilic, and the tails of the molecules, phospholipids, are hydrophobic. And those two, it won't allow it to pass through. So a transport protein is going to act as a channel that's going to allow these larger molecules or molecules that can't go through the membrane to pass. It is still going down the concentration gradient. It is still high to low concentration. Examples would be glucose. That's our scientific term for sugar. It's a large molecule. And then we have sodium, usually ions, which is like salt, Na pluses that will be moving across as well. Oops. So here's sort of an example. You have a membrane, you have this protein here that has an opening. You still on one side have a large number. It wants to get through on the other side so that it is balanced out over time. Facilitated diffusion, same as diffusion, no energy, has a molecule that will aid that. And then we have osmosis. It's a special type of diffusion. And what they don't mention here is water actually has to have its own little protein that allows it to go through. It's called an aquaprotein because it can't go past the phospholipid tails. But osmosis just deals with water, the diffusion of water. So this is when specifically water molecules are going down a concentration gradient from high to low, because sometimes what's in the water cannot go across for whatever reason, but the water can go across and kind of balance out the concentration. So so over time, and notice the water level changes. The little dots in there doesn't change, but the water changes. Caleb, if you can't, just
So we doing something
stand
with
right
the
next. Go get you some water or something. You gotta wake up. Yes.
We doing something with the other guys?
Yes. We'll go lab in just a minute.
You want to eat some?
I might let you eat some. All right, so osmosis is a little more complicated because we're looking at the solution of the substance. So when we are looking at the solution, we're looking at the amount of solute that's in that solution. So hypertonic solution means that it has a lot of solute. So the water concentration is less than what would normally be found in cytoplasm. And so as a result, water is going to move out of the cell, and the cell is going to kind of shrivel. Hypotonic solutions, water concentration is above. All right, so this is the solution outside of the cell. Let me back up to the first one. Outside of the cell, there's less water than inside of the cell. So water will leave the cell and come out. Hypotonic is the reverse. You have a higher concentration of water inside the cell than outside. So the water concentration outside the cell is lower, and water is going to move into the cell, and it's going to swell. Isotonic means it's the same. So, and the cell's not gonna shrink or shrivel up, it's gonna stay the same. So I try to think about this with IV solutions. If you went to the hospital because you were dehydrated and they just put an IV of pure water in your cells, that means the water concentration outside of your cells is higher, and so water is going to actually move in and your cells will swell and it would cause a major problem. So they can't, when you go to get IV for dehydration, they're not just giving you pure water. They're giving a mixture of water and electrolytes that match what is the concentration that's in your cells so that your cells stay the same size, but yet you're getting the electrolytes and water you need to become undehydrated, to become hydrated again. So that's sort of a real live situation. So you wouldn't want to give it too salty of a solution with too much electrolytes, because then your cells would shrivel, and that's bad too.
I wasn't done with that.
Oh, sorry. Back up. Taylor, you gotta wake up. Gotta do the work, you gotta get that grade done.
Miss Noel.
Yes.
So for the lab, do you want me to just get a piece of notebook paper?
Well, I have I'll show you that assignment in a minute. There'll be something where you can record your information, but you'll just have to record, like I don't think we're measuring anything today, but if we are, yes, we will be measuring something. So as we're measuring, the one that I do, you'll just write that data down so that you'll have data. And I'll zoom the camera in to what we're doing up here on the table when we get to that point. All right. So this diagram here is going to come in handy when we do our practice a little bit later. So hypertonic, notice how big the H2O is. There's a lot of H2O in the middle. And we're going to see this with our gummy bears in the next day or so.
Is there anybody on
Yes.
there? How am I gonna get over there?
You just walk across. You don't worry
What am
about
I waving at?
it. Amber.
Hi.
Outside, notice how little the H2O is written. You are not, that means a little water. So the water is going to move out to where it's smaller. So in this case, it's going to shrivel. This case, hypotonic means you've got more water outside. Look how big the H2O is, how little it is on the inside. Look at your arrows. The water is going to move in, and your cell will swell. So that would be the example of getting pure water as an IV. Your bodies would swell, or your cells would swell till they burst. And isotonic, notice they're the same size here. Water's just going to move in and out, in and out. It's going to stay balanced. And no change in the cell. So this right here, I don't know if that's in your notes. I don't have a copy that I'm looking at right now. That is going to be a reference you may want to come back to when we go in the slideshow when you get your practice sheet to work on. We're stopping there with the notes. Let's split this up over 3 days here. All right, so what we're gonna do next is we're going to look at this lab. So I put a little microscope here, and we've got a lab that we're gonna explore with osmosis. So this is what you would click on, Amber, so you can see the sheet. And I'm gonna give everybody here one.
I looked at the rest of them.
Yes, if you can go quick. All right, so for this, if you look at the guiding question, how will the size of the cell represented by the gummy bear change in different types of water? All right, we're going to investigate the movement of water into and out of a gummy bear. So you're going to make predictions. So in this first part, we're going to use tap water. I have a little bit of distilled water. I hope I have enough for everybody. And hopefully I still have some salt. And then we're going to make some salt water. So you're going to make a prediction here of what's going to happen to the gummy bear in each type of water. So on your paper, you're going to, in making your prediction, you're going to tell me, is the gummy bear size change, stay the same? If it changes, is it going to shrink or is it going to swell? So I would write each type of water. So you would say tap water, distilled water, salt water, and tell me what you think is going to happen to that gummy bear. You do not have to be right here. So what I'm going to do is I'm going to stop presenting because I want to be able to see myself up here or on my other screen. So stop presenting. Thought I could see myself on this other screen. I guess I'm not going to see myself on the other screen. Yes, I can. I can pin it. So I won't be able to see what I'm doing here. So if I want to get this focused in on this table here,
That's neat.
I can kind of see. All right, so I've got distilled water. I've got cups that we're going to use. We're going to make some salt water. And we have gummy bears. So I'm going to give everybody 2 cups or 3 cups, and you're going to write your initials on the cups. And trying to think what I'm going to do here. Let me look at the directions first to make sure I'm doing this right. So we have plastic cup, tap water, distilled water. All right, so for today, we need tap water and distilled water. You're going to measure the mass of each gummy and record it in the table. And then you're going to place a bear in each cup. You're going to fill the tap water cup with tap water. You're going to fill the distilled water bear cup with distilled water. And then we need the same amount of in each, and we're going to let the bear sit for 24 hours. All right, so you're going to need 2 cups. I'm going to need something to measure some water so that we have enough. So we're going to need, I'll give each of you, if you will put tap water and distilled water and your name on each cup, and see if it'll write on the plastic. Oops, you need 2 cups. And, all right, tap water and distilled water. You could put T water and D water if you wanted.
What's distilled water? Like,
Distilled water has taken anything out of the water. Natural water has some minerals in it, and so it's made sure that everything has been taken out of it. So
Tap water, what?
distilled. So I'm just putting tea water, and it's, you won't be able to see it in one. And I'm putting D water. And then you're just gonna put your initials. But make sure you have made your prediction. So under hypothesis, with your pencil, you needed to have made a prediction. What's gonna happen to the gummy bear in tap water? What's gonna happen to it in, distilled water? What's gonna happen to it in, salt water? But we're not gonna do salt water yet. So, you should have your cups labeled. And then the directions said to measure the mass. And I've learned from past experience, my cheap little scales do not work. We have to use the triple beam balance. Does anybody know how to use the triple beam balance?
Well, that,
Remember?
Yeah. Yeah, that's what I think it is.
So, I'm going to demonstrate with mine, and then each of you is going to come up and weigh your two gummies. Do you think the gummies will be the same weight?
Yep.
Well, let's see. So I'm going to put, I like using the colored gummies, or the ones in there that are white don't usually show up very well when we do this. So, and Amber, you'll put down this information. You see the chart on the second page?
Yeah.
That's where you're gonna put, initial day. So we're going to put, tap water and then distilled water under initial day. So when you're using the triple beam balance, trying to get where everybody can, at least you can see Amber, but those in here can kind of see. Make sure everything is on 0. You're gonna start moving the smaller one, because these are gummy bears, they're not that heavy. And what did I forget first? I didn't zero it out. These things are never on 0. All right, so blank. Alex, you can look at that better. Is that line showing up right on the 0 there? You or Caleb one?
I can't-
Yeah.
I don't know what to think.
Look, right here. So Amber, you can see it? Is that pretty good on the screen?
Yeah.
Oh, I'm zoomed in as much as I can. So it is zeroed, so I'm gonna put my gummy back up. I'm gonna use this bottom one. I'm gonna just move it. It went past the line, so I'm gonna back it up. Kinda like to use a pencil.
Yes.
Mhm.
It's like one of them doctor scales.
Yes, they take your weight on, right? All right. Oh, I think I did it. Okay, so it's back balanced. The line is on the 0 here, so I'm gonna try to read this upside down. It went past the 2, and I'm on the 1, 2, 3, 4th mark, so that's gonna be 2.4 grams for mine. You don't write that, because each of you is gonna measure your own. So my green gummy is 2.4 grams. And then I'm gonna measure my red gummy, and I'm gonna cheat a little bit. I'm gonna see if it balances by just putting it up here. In this case, it did. So both of my gummies are actually the same size. We have found that they are, in the past, they haven't been the same amount. So for me, my two gummies, so I'm gonna use the tap water. It's gonna be my green gummy. So Amber, that's what you will do. My distilled, and I think I'm gonna spell distilled, is going to be red, and it's also 2.4 grams. Now, to conserve water, you're gonna measure it. You're gonna take your tap water, you're gonna take a beaker, and I'm gonna tell you how much in just a minute. I gotta make this a little bit of distilled water work for all of us. So, I think we're gonna use 50 milliliters.
You drink that?
Yeah, my husband chooses to drink distilled water. Well, I don't know. All right, so I've got 50 milliliters. So in my distilled water, I said my red one. And I'm going to, I've measured my 50. I'm going to pour it in. And then I'm going to set it to the side. I'm going to take this other beaker and I'm going to go to the sink and I'm going to fill it to 50. Hold on just 1 second. Let me get the rest of these instructions and then you can go. You may have to set it up here on the shelf, make sure you're right at 50. And you're gonna put this in with whatever color you choose for your tap water. And then we're gonna set these to the side. So that's what you're gonna do. So each of us, so if you'll make a note, Amber, we did 50 milliliters of water. Well, that was for your hypothesis on the front side.
All right.
So tomorrow, we're gonna check these, we're gonna weigh them, and then we're gonna do part 2. And I think there's actually a third part we'll do on Thursday. So we're going to, right now, we're looking at the difference between tap water and distilled water. So each of you are gonna take a turn to come up, measure, get your water, and put this in your cup. You need to have your hypothesis on the front page, and then just your only thing filled out on the second page is your weight for the initial day. All right, who wants to be first, and who has to go to the bathroom? Do you want to do your gummies first, or do you want to go to the bathroom first? Okay.
It was
All right,
52
who's come up first?
milliliters.
You have a question, Amber?
It was 52 milliliters of water.
Yeah, it was 50 milliliters of each type, so 50 tap water, 50 distilled. And then Amber, you could go on and look at the next assignment.
Do I press turn in on this one?
Huh?
Turn it, like, do I press submit on this one?
No, this one we're not gonna submit till probably Thursday, or you might have to finish something up Friday, or we'll finish it up Monday. This one will stay open.
All right.
And then there's 2 videos and then a practice that uses your notes, I think. So you can work on that. And I'm gonna stop recording right now.
All right.
Feel free to stay on. You can watch them measure.